It gives me a great pleasure to welcome Hinrich Stecker, who's going to speak on the future therapies for inner ear disease, progress and possibilities. He is a full professor at the University of Kansas, fantastic researcher, has worked in this area for quite a while, great friend to House Institute. And so Hinrich, thank you for joining us, and we really appreciate your time giving us this lecture. So thank you.
Thank you for that kind introduction and the invitation. So there's always a number of different things we can talk about. So what I've done over the really past 20 years is work on gene therapy for the ear. And I wanted to take the opportunity, instead of focusing just on the molecular biology angle of it and why, to try to put this in a broader context. So we all know that hearing loss is important. So this slide shows the annual cost of untreated hearing loss, and obviously we're very focused on our patients in the United States, but the World Health Organization has recognized that really it is a huge problem worldwide, costing billions of dollars in lost productivity and suffering. And recently also recognized that hearing loss, untreated hearing loss, is one of the major risk factors for developing dementia. So obviously, treatment of this is something that we've all dedicated our lives to, and I think it's important to really highlight what an important role we as otologists play in the care of these patients. So I want to start out with really highlighting these three gentlemen here. Obviously, the development of otology involves thousands and thousands of clinicians and researchers, but I wanted to sort of start out by making a point of three individuals that really changed the direction of otology over the last 200 years. So the gentleman on the left is Schwarze, who is the professor at Halle, who really codified and developed mastoidectomy for suppurative otitis media. Really, in his age, people died regularly of mastoid disease, and he was really the individual that published the first very organized study of mastoidectomy. In the middle is Julius Lempert, who developed the fenestration operation and really brought otology into the era of correction of hearing loss, focusing initially on conductive hearing loss, which was largely allowed by the development of antibiotics. And obviously on the right is Bill House, who is instrumental in the development of neurotology and really moving otology into the treatment of neural disorders with his development of the cochlear implant and development of surgery for acoustic neuroma. And obviously, the House Ear Institute has played an incredibly important role in the dissemination of information and training of generations of physicians. What's less appreciated, and what I probably want to point out about the House Ear Clinic is what an important role the development of instrumentation played really in the development of our specialty. The really broad use and development of the surgical microscope, the development of tools for surgery, and a lot of this is really going to be needed to move otology into the next era. So hearing loss is a vital sense. I wanted to point out with this slide with these creepy crawly things here, there's plenty of blind organisms, but there's really no organisms that can't orient themselves to gravity and that don't hear. Those are lethal mutations. So hearing loss is a sense that is tied into the whole organism. Yet, interestingly, even though this is such a vital sense, we have not developed any pharmaceutical treatments, including small molecules or up to cell therapy, that really can be applied for hearing loss. So what are our options really for the design of inner ear therapeutics, and how do we deliver them? Really, what I'm going to focus on a little bit in my talk is the use of gene therapy, which you see in the illustration on the right here. It's our ability to package genes of interest into a virus that's been engineered to be non-pathogenic, get that into a cell, and then change the phenotype of that cell. What we can do with this has been hugely expanded by the Human Genome Project, which has given us an understanding of many of the genes that are involved in the pathogenesis of inner ear disease. So we basically have three options for developing a medication. We can think about the rescue of genetic hearing losses. We can think about the protection of existing structures, so preventing hair cell degeneration, preventing degeneration of the spiral ganglion, or we can think about the restoration of previously damaged structures. So when I started my residency, this was sort of the conceptualization of what the inner ear looked like when you look through the stapes footplate. So you could open the stapes up. You didn't go inside at all. The first cochlear implants at that point had been done in profoundly deaf individuals, but really, 30 years ago, the ear was really considered still an environment that could not be instrumented. All of that really has changed with hearing preservation cochlear implantation. The audiogram on the right shows the pre and post-operative hearing in one of my patients that was implanted with a 28 millimeter long Med-L electrode. So we have the capacity really to open the inner ear, place something really rather large inside, and keep residual function. This by extension also means we can inject drugs, we can put devices in that deliver drugs, and this really expands our ability to treat patients. So what do we pick? How do we get started? Obviously, every big drug company is interested in a cure for presbycusis. It's very common. They can make billions off it. The problem is presbycusis is a mixed pathology, and it has a very long, slow progressive time course. So just the logistics of building a clinical trial and following patients for years and years probably upfront makes this a bad choice for a clinical trial. Sudden hearing loss has been popular. It's a low-risk population. We have little to offer these patients upfront other than steroid therapy. But again, if you really look at this, it's a mixed pathology. It's relatively rare. We don't understand the mechanism. So you really need a very, very large number to power a clinical trial. Spiral ganglion dysfunction and augmentation of spiral ganglion function with neurotrophins has been a popular topic. That's actually what I did my PhD thesis on. And protection of residual hearing in cochlear implantation. The pros of this, we have lots of different molecules to choose from. CI patients are a great trial population because we are accessing the inner ear. Downsides are we potentially need long-term delivery, and there are still some problems with picking good outcomes measures given the variability of cochlear implant speech outcomes. There are also regulatory issues. A lot of the manufacturers are afraid that if we combine a drug with a device, it could lead to complications of the device and their device being taken off the market. What I've sort of focused on in the last 20 years or so is hair cell loss. The pros of this, we have a very defined molecular pathway that is well described. We know how hair cells are generated and potentially can be regenerated. The good things about animal models is when we build a model with hair cell loss, we can count very easily, are the hair cells there or not after our treatment. The downside to hair cell regeneration as a trial population is it's, and I'm going to get into this in some more detail, diagnosis is difficult in our initial trial populations. Ototoxicity is a good population for studying prevention strategies. There are defined patient populations, multiple different molecules available. In many cases, delivery is difficult, and in the case of cisplatin, you have to worry about the molecule that you're testing interfering with the anti-oncogenic effects. Finally, genetic hearing losses. This is steadily moving into the clinic. The advantages, we have a defined molecular diagnosis that we can treat. The disadvantages, many of the genes that cause sensorineural hearing loss are outside the size limits of available vector systems. A large percentage of these disorders present prenatally with degeneration of the organ of Corti. So from an ethics standpoint, it's difficult to organize a trial. And many of the disorders are dominant, which is much, much more complicated to treat than a gene replacement strategy. So what you really want to do when you are looking at all of this is to reverse engineer the whole situation. Determine a target. You have to understand the regulatory environment. What are we going to be allowed to do? Pediatric trials are harder than adult trials. There has to be a definite benefit to the patient. You have to have a good animal model. And I would argue that in many cases, mice are not reflective of what's going on in our human patients. You have to have a population of patients available. If you're building a therapeutic for a genetic deafness and there's only one family in Morocco that has this, even if it fits in your vector, and you have an animal model that shows good recovery, you're not going to be able to build a successful clinical trial on top of this. Okay, so into hair cell regeneration now. Our group have focused on the use of the master switch that controls the genesis of hair cells, called atonal, to regenerate hair cells. So atonal is a transcription factor. That means during development, it turns on the switches that make a cell a hair cell. It's present really in all vertebrates, and a form of it is actually even present in Drosophila. And we know that if you knock the gene out, mice do not develop either auditory or vestibular hair cells. There is really a wealth of studies that show that if you overexpress atonal in a damaged auditory or vestibular system, you can make new cells that look like hair cells, and you can recover at least some auditory function and definitely some vestibular function. So based on this, we developed a gene therapy vector that could deliver the human atonal 1 gene. It's called CGF166 for the clinical trial. And our hypothesis was if we delivered this to a damaged inner ear, we would force the surviving supporting cells to become a hair cell. This is what the molecular biology picture actually looks like. We used a type of virus called an adenovirus, which is a virus that causes common colds that's been modified not to cause disease. So the viruses E1, E3, and E4 gene regions are deleted, and then we put in the human atonal gene and a promoter, which is a switch that turns on the gene that it is next to. So all of this is packaged, and there's actually a lot of background work that goes into validation to make sure that all the pathogenic sequences have been eliminated and that you have a very pure stock of vector that can be injected into people. So our clinical trial has, our phase 1 clinical trial has finally closed. I, because of Novartis agreements, can only present some of my data. So our initial entry criteria really looked like classic cochlear implant patients. So we wanted patients that had some hearing left, so we could test if there was side effects of the vector in terms of hearing loss, but patients that really had such terrible hearing that we wouldn't ruin residual hearing. And that was sort of initiated by the FDA. My initial conceptualization of this project really was to target it at gentamicin vestibulotoxicity patients. But most of them actually have a lot of residual hearing. So this is our best performer patient. This is the before and after audiogram. We got some recovery of hearing in the middle and low frequencies. And this right here, and I'm going to come back to this, looks at the 15 patients we did at KU. The volume of viral vector that we infused and the number of patients that we tested, and this is actually important for the later discussion. So so we had 2 responders in the whole trial and a lot of non-responders. So a big question comes up is, how do we really improve outcomes, and why did the trial end up the way it did? And basically, we can sort of divide it into 3 areas. Delivery is important. The actual vector you're using is important. We can do things to the virus that we deliver to make it more efficient. And probably I'm going to spend the latter half of the talk on this, is we really have a deficit in diagnosis. So how do we actually decide, and how did we decide how to give this? In humans, and I'll show this on the next slide, we have a couple of different ways to get in the inner ear. In our mouse animal models, especially rodents, rats, and mice, it's very difficult to deliver into the cochlea directly without wrecking the middle ear, not being able to test hearing. So for rodents, all the delivery is done through injection through the semicircular canal. In the monkey studies we could inject through the stapes footplate or through the round window. And for humans, we decided that we would inject through the stapes footplate because this was the most direct route. So in humans, we have a couple of different ways we can inject into the inner ear. Stapidotomy has been around as an access point to the inner ear for the longest time. Everybody knows how to perform this. It does not take a long time. It's a very direct approach to the inner ear. And importantly, it's far from the cochlear aqueduct. Cochleostomy, round window injections, we base this on cochlear implantation. It gives us access to the scala tympani. To do it properly without designing special insertion devices really requires a facial recess, and it takes longer. And the FDA opinion was that for a very, very high risk initial clinical trial, we should take the most direct surgical approach. Finally, there's also data out that you can actually deliver via the endolymphatic sac. We're going to spend very much time on that today. But Vittorio Coletti showed years ago that injection of gadolinium into the endolymphatic sac actually results in delivery of gadolinium all the way to the apex of the scala media. So that's a possibility as well. An important thing the FDA was really focused on was where does the vector go? And a continual question is always, what is the patency of the cochlear aqueduct? You can see it very clearly here on the CT scan. It is right inside the round window. So if you inject here and you have a patent cochlear aqueduct, some of your drug is going to go into the CSF space. There's also data from a number of different papers showing using tympanometry methods to demonstrate that really the cochlear implant, the cochlear aqueduct is patent in most individuals until much later in life. And all of you who've done a stapidotomy and seen fluid slowly well up from your footplate fenestration, those patients have a patent cochlear aqueduct. So there's also a really great temporal bone collection in Uppsala, Sweden. Helga Rask Andersson has a huge collection of temporal bones that have been injected with plastic and all the bone corroded away. And I actually went and looked at those, and you see in most of them, when you look, the cochlear aqueduct is really not a single duct, but can be sort of seen as open strands that the plastic that was injected through the oval window has gone down. So it's really patent in most individuals. So because of that, we developed a system, a micro pump that delivered very slowly, a needle system that could be balanced on the footplate with a little stopper that allowed the needle not to be introduced into the ear too far, to deliver through the stapes. What I never realized when I sort of started this is all the work that goes into this. Even the packaging had to be tested for vector adherence. Close to $1 million were spent putting together the sterile kits with the syringe and infusion tubing and needle, and all of that has to be produced for the clinical trial. I've shown this at some meetings. This is a photo of what it actually looks like. Again, these patients have a mobile footplate. Use a CO2 laser to make a tiny little hole in the footplate. This is the most controlled way of doing it, and can then put in your catheter. And again, I want to go back to, again, the house ear, all the innovations in devices and instrumentations that have come out of house. You can see this is sort of jury rigged and developed. This is not a mature technology for delivering drugs to the inner ear. Really, a ton more work needs to be done until we mainstream these sort of disorders. Go to the next slide. Another thing that needs to be taken into account are design issues. I'm going to get into the molecular biology of things a little bit. Besides the gene of interest that we're putting in, things like promoters, the gene switches that actually control how much of the substance we're making in a cell are important. And this is a study done by Mark Pretorius, who was in my lab for a while, that tested the ability to regenerate hair cells using different promoter systems. The CBA and the CMV promoter are widely used in gene therapy experiments today, but give a much poorer outcome than the promoter that we actually ended up picking for our clinical trial. So all this work needs to be done up front. This is some animal data as well. So this is a mouse utricle showing a regeneration of hair cells. The hair cells are seen in green. So an untreated utricle is seen here, a utricle that's been treated with aminoglycosides and most hair cells destroyed is seen here. And then you see two different doses of an adenovirus carrying the human atonal gene. And the takeaway message here is that gene therapy, just like any drug, just like Tylenol, has a dose response. We need to get to a certain number of particles of this vector carrying the gene delivered before we see a hair cell regeneration effect. And we can define that number very clearly. Based on this, we can then move to scaling studies. So we need to know if this works in our mouse, how much do we have to give in our human patients in order to estimate that we're getting an effect. So based on our animal data, we knew that the mouse perilymph has a volume of about 2.5 microliters, and we need to give about 5.5 times 10 to the 7th vector particles to see hair cell regeneration. We see our best effect at around 5.5 times 10 to the 8th. We can then go through different size animals and develop a scaling. The bigger cochleas obviously need more vector injected into it. And when we do this in humans, we see that really to start to get into an amount where we would expect to see hair cell regeneration in the human cochlea, we would have to deliver about 80 to 90 microliters of concentrated vector. Our trial maxed out at 60 microliters before we started seeing some hydraulic effects and loss of residual low frequency hearing. So we actually never got probably to an effective dose of vector in most of our patients. Again, back to the concept of better devices. I really think the future in delivery lies in having better ways to get drugs into the inner ear. The illustrations at the top show the Med-L Cochlear Catheter. This is essentially a hollowed out cochlear implant with a delivery port at the end that can be inserted really up to 20 millimeters into the cochlea. And in this sort of dye study, looking at different insertion depths, you see how differently drug would potentially distribute with deeper insertions and deeper delivery into the inner ear. So my study basically injected into the scala vestibuli, but probably never reached the middle and apical turns in a very, very high concentration. Even with that, we got 2 patients that had some recovery. This device has now been used in humans. It's CE marked in Europe, and this is Niels Prenzler, who published a very nice study on preoperative delivery of steroids into the human inner ear prior to cochlear implantation. We can also make our vectors better. So we use sort of an off-the-shelf vector that had been used in the eye, and we've developed vector systems that are more efficient and for the same number of particles give you a better hair cell regeneration outcome. Finally, what we're missing is pathology. So we're looking at patients that sort of have this configuration of hearing loss or a little bit better. And if we look at our temporal bone collections, you'll see often hair cells are not the problem in these patients, but we really have no good way of knowing that ahead of time. Also, probably in our severe to profound hearing loss populations, we underestimate the amount of genetic hearing loss that is present. So all patients were screened for family history of hearing loss, but if you look at a recent study from Japan, where they sequenced a whole population of cochlear implant patients in adults, they find 20 to 25% of patients undergoing cochlear implantation for severe to profound hearing loss actually have an identifiable genetic disorder without any family history of hearing loss. So if you take all of this together, we have problems with delivery, and we probably do not know what is wrong with our patients when they come in to see us. So we do have efficacy in our preclinical models. We can deliver safely. We found out from our volume delivery studies that we can probably deliver about 10 microliters per minute for a total of 30 microliters with a single entry point into the inner ear without causing hydraulic trauma. And this was probably quite a bit below our estimated maximum drug needed for efficacy. So hopefully later versions of the trial will be more targeted and take some of these factors into account. So what are really our next steps? So really to optimize treatment and develop new treatments, we need to know what the structure is in the inner ear at a time. We need a physiologic measure of what's going on in the inner ear, and ideally a molecular diagnostic. At the moment, clinically available, we have otoacoustic emissions, we have ECOG that really measures the initial depolarization of the ear and gives us potentially some ideas about things like synaptopathy. We have the pure tone audiogram, and we have ABR. Otoacoustic emissions are really the only tests that give us cellular-based information about what's going on in the ear, and they really do not work in any hearing loss beyond a moderate sensory neural hearing loss. An interesting study by Tina Stankovic that looked through the mass eye-near temporal bone collection, looked at the sort of architecture of the audiogram and found that the audiogram itself did not predict the cellular basis of hearing loss based on regular light microscopy. Charlie Lieberman has published a follow-up paper that showed that with some newer microscopic techniques, this could be refined a little bit, but basically when you have somebody like this walk into your clinic, you can't really with any accuracy say this is a hair cell problem, this is a stria problem, this is this is just a molecular problem in within the hair cells, but the hair cells are still there. So basically, we have no pathology. All of oncology now is guided by a molecular analysis of what is going on in the patient. And to really move the field forward, that's where we need to go. The last couple of years, my lab has focused really on looking at developing better biomarkers for various types of hearing loss, so that we can really improve the patients that we pick for our clinical trials. So when we look at biomarkers, there are sort of a couple of levels of molecular biology that get us from our genetic blueprint with the DNA to the phenotype, what's actually going on in the tissue. So basically from DNA, we make an RNA copy that's made into protein. That's referred to as transcriptomics, and the measure of the proteins in a tissue, or really measure of the proteome, and the proteome is then modified by glycosylation and modification of proteins to make enzymes and have a metabolic effect. So all of this interacts with the environment, and really biomarkers can be derived from any level of this process. We are at the moment in hearing most familiar with genetic testing. So there's been some really interesting studies coming out on cochlear implant patients showing that genetic variants can really predict outcomes of cochlear implantation. And of course, we're all familiar with the follow-up of infant hearing screening with genetic testing to identify disorders such as mutations in connexin 26. So probably the next thing to go to clinical trial will be a clinical trial of gene therapy for otoferlin mutations organized by the company Akuyos. And again, this is identified by physiologic testing and identification of auditory neuropathy and follow-up genetic testing to really ensure that your patient population is appropriate for the therapeutic. A lot of work on proteomic sampling of inner ear fluids has been done. This is Atanaja Warnecke at University of Hanover, who's done a lot of the seminal work on this. And she has some papers out now really showing that she can measure inflammation in the ear and sort of start to predict the outcomes of cochlear implant patients based on the inner ear proteome. Finally, metabolomic sampling has also been carried out in cochlear implant patients. And based on the metabolome, you can actually tell the duration of hearing loss in patients undergoing cochlear implantation. My lab has focused on microRNAs. So microRNAs are short non-coding RNA molecules that are used in the regulation of gene expression. So microRNAs basically bind messenger RNA and cause a breakdown of messenger RNA. And the reason microRNAs are interesting is they're very, very robust. So they get into all kinds of different body fluids. And for example, you can actually find microRNAs related to glaucoma in the serum. So it makes it pretty good that microRNAs are potentially markers for inner ear diseases too. What we've done is sample perilymph from cochlear implant patients, from stapidotomy patients, from the occasional patient undergoing labyrinthectomy, and use a gene chip technology to really identify which microRNAs are in the inner ear. And this is work done by Matt Shu, who's now in fellowship at WashU. And basically what we do is we attach a colored label to all the microRNAs we find in the perilymph, and then hybridize them to an array. And one of these arrays has about 40,000 different microRNA types that it can identify, of which about 6,000 are human. So from each perilymph sample, we can identify about 6,000 different microRNAs. There's software packages that are involved in the analysis of this. And basically, we can then look at the microRNAs that are occurring in the perilymph and look at genes that are expressed in the inner ear. And we've built some libraries of human genes that are taken from patients with normal hearing that are undergoing transcochlear surgeries. These are rather rare. And then kind of make a guess. What are the microRNAs doing in the inner ear? So the microRNAs that we find are going to bind different messenger RNAs in the inner ear, target them. And so by looking at this, we can make guesses about what they are doing in the inner ear. I had to put in one picture of Helena. This is Helena operating with Jim Lin, whom you may remember. Helena did a lot of the math number crunching on some of these early studies, and we have a couple of different ways of looking at this. We can pick a microRNA that's responsible for a pathway that we're interested in and look at this. We can use mathematical techniques such as cluster analysis or advanced techniques such as machine learning to really sort of start to look at what some of these little RNA sequences may be doing. So this is what this actually looks like. I actually started out by reading Bioinformatics for Dummies to get sort of a handle on how to look at this. But this is 28 of our patients initially expressing 6,000 different microRNAs. The list is incomplete here. At different levels. Blue is low, red is high. You can then use software. This is a software package called ClueCore that takes all this information from each individual patient and puts it into one point on a three-dimensional graph. And by how far these points are away from each other, you can decide how similar a patient's microRNA profile are. And I don't want to get into the deep details of it, but basically there's 3 disease populations shown in different colors here, and you can see sort of the pink group segregates away from the other groups, and we can then go in here using software and decide what are the factors that makes this group different from that group. So what comes out of this are a couple of different microRNAs, and we've now picked a couple that differentiate 2 groups. This is from a Meniere's disease study, and then we can look at what genes in the inner ear do they actually target, and based on that we can look at these genes and see where they're expressed in the inner ear, and sort of come to a conclusion about what the microRNA profile is telling us about pathology in different cells within the ear. So this is all gets kind of complicated, but we can use different techniques that allow us to look at huge samples without getting bogged down in 6,000 variables to really find key markers that differentiate different disease types. So machine learning is used by Google to decide how long it is going to take you to get to work, and by Facebook to decide what ads to send you, and is available now in publicly available software packages that Matt Shu started this out, used to really look at whether microRNAs could predict which patients had residual hearing or not. So looking at a population of patients with profound hearing loss, with normal hearing undergoing stapidotomy, and with sort of moderate hearing loss undergoing hearing preservation cochlear implantation, he was able to really identify microRNAs that separated residual hearing patients from non-residual hearing patients, and then actually even break this down further and identify microRNA populations that could group patients really into 20 dB hearing loss cohorts. So based on your microRNA profile, and this is in this paper, we can really decide whether you have no hearing loss, mild to moderate, moderate to severe, or profound hearing loss without seeing the audiogram. So that's where we're at with that. So I think this is going to be really vital for planning further clinical trials. We need to know exactly what's going on in the inner ear to deliver a therapeutic. I wanted to make sort of two more points again on delivery. I talked about it a little bit, how we need really better delivery devices for the inner ear. A lot of our preclinical modeling is based on rodents. So this shows delivery of a drug in a poloxamer gel into the guinea pig inner ear. The problem is the guinea pig and the mouse is much smaller than the human. And when you place something at the round window, probably you're not going to get delivery all the way to the more apical regions of the inner ear. So you're going to need a device really to get that drug there. And this is from some of Alex Salt's work. So I think we as surgeons shouldn't be afraid of this next step into molecular therapeutics. We're going to be needed to both sample the inner ear to figure out what's going on, and needed for a whole new generation of operations to deliver drugs to the inner ear. I would like to point out this paper too. This is a great paper from a number of different labs that came out in hearing research that really looked at human temporal bone pathology to analyze what the issues are with delivery to the inner ear. And the conclusion they came to is really that middle ear based delivery is probably useful for some limited things, such as delivery of gentamicin, but really to broadly treat hearing loss throughout the inner ear, we are going to need a different strategy. Up and coming, and I'm going to mention this only very briefly. Again, I said at the beginning that cochlear implants were sort of a great gateway into developing drugs for hearing loss. There are a number of systems out, the catheter I talked about, but there's also cochlear implants that have delivery ports that can be combined. The first combined drug device electrode is currently in clinical trials. So I think 3 patients have been implanted at Hanover with an electrode that has dexamethasone-eluting strips built into it to sort of protect the hearing. So I really think this is going to be the way forward to look at patient populations where we can use the cochlear implantation process for patients that are about to undergo cochlear implantations to test novel therapeutics. So I think experimental design and model identification is important. We need larger animal models, such as a pig with a large volume cochlea, where we can really look at how drugs distribute. The mouse is not adequate. We have lots and lots of different agents that we can potentially consider. ARO meeting has been going on for years and years, and there's thousands of different molecules suggested at each meeting. I think we've shown that we can at least safely deliver into the inner ear, but that a lot of different things need to be considered to really put together a trial. And finally, we really need to develop better tools to access the inner ear, both for diagnosis and treatment. So I'd like to sort of come back to my first slide again. What these gentlemen on the left really did is assemble ideas from multiple sources. They published and taught that really allowed dissemination and testing of their ideas and replication of their ideas, and allowed really very novel and radical thoughts to reach the mainstream. I mean, for each one of them, the procedures they proposed was a radical departure during the time that they were in practice. And probably most importantly, to get us here to the future, we need to go back and see what steps these giants in our field took to really get the field to hop to the next level. So you have to study and importantly, be willing to take a risk and a step into the unknown. I'd like to finish by thanking everyone. Really unbelievable number of individuals have been involved in this. This is sort of my core work group right now. I had a great collaboration with Novartis, who really turned this project into a clinical trial. My long-term collaborator, Doug Bruff, who's a genius at construction of adenovectors, collaborators Charlie Della Santina at Hopkins, Larry Lustig at Columbia, Chris Runge at University of Wisconsin, Atanasia Vanica at Hanover, and of course, Matt Shu at WashU, and Helena, who is now with you. Thank you very much.